I hope that you won't be disappointed, and please interrupt me if you want at any time. So today, to end up this, close this week of talks, I'm going to talk about another highly debated subject that is quite controversial, and there are many different ideas on stake, and a strong opposition between different models, as you will see. And that is the question of the origin of eukaryotes. And so to try to dissect and try to clear up things, I will talk first about the facts, the observations that we have, and the models that try to explain the observations that we have concerning the origin of eukaryotes. I will then comment on how we can perhaps hope to test these models, and I will end up by discussing some really very open questions that will be the focus of future research and certainly speculation in the future. So, first, the facts. There are two major structural types of cells on Earth. The prokaryotic cells, where you have the DNA, the genetic material, directly immersed in the cytoplasm of the cells, but there are not clear intracellular membrane structures in general, and the eukaryotes that have an endomembrane, a complex endomembrane system, defining, among other things, a nucleus that contains the genetic material, the chromosome. So the concept, the term eukaryote was introduced by Chaton in the early 20s, but the modern meaning of prokaryote and eukaryote was really due to Stanier and Paniel in the 60s. So we have two types of structures in cells, but we have, as we have been discussing during the week, three different phylogenetic domains of life, two prokaryotic ones, the bacteria and the Archaea, that resemble morphologically and have many different metabolisms, and the eukaryotic domain that has a structure that is eukaryotic. So, in the case of eukaryotes, most of them are composed, most of the diversity of this domain is composed by unicellular lineages, but there are several lineages that are multicellular or contain multicellular organisms, suggesting that multicellularity has evolved independent times in the eukaryotic tree. So we have seen that already, but it is good to indicate it as well, because it is important. Now we have another third observation that is also important, and it is that prokaryotes, Archaea and bacteria, predate eukaryotes in time. So there are two types of evidence for this. One is fossil, the fossil record. Even if the very ancient fossil record is more or less doubtful, we know that we have stromatolites, fossil stromatolites, dating back at least at 3.4 billion years ago, and at 2.7 they were really widespread and massive. Some people think that they can identify prokaryotic microfossils in this kind of structures, but the most serious evidence for the presence of prokaryotes at that time is that around 2.4 billion years ago, and certainly at 2.1, there was a massive increase of oxygen in the atmosphere. And this oxygen is the byproduct of oxygenic photosynthesis. And oxygenic photosynthesis has only evolved once in cyanobacteria, and all the plants and all the eukaryotic algae have inherited this capacity, photosynthetic, oxygenic photosynthesis, from the endosymbiotic cyanobacteria that now are the plasts. So this means that cyanobacteria must have been evolved much earlier, 2.4, because oxygen had already begun to accumulate in the atmosphere, and before it can happen, it has first to oxidize all the minerals. So all the minerals at the surface had to be oxidized before oxygen could accumulate in the atmosphere. That means that most likely cyanobacteria had already appeared at 2.7 billion years ago, when massive microbialites, fossil stromatolites occurred. Okay, so this is fossil evidence for the existence of prokaryotes. Furthermore, one additional point is that cyanobacteria is a derived group of bacteria, so it means that bacteria have already diversified. Okay, so it means that most likely bacteria evolved much earlier. And by contrast, the oldest non-contested fossil eukaryotes, we don't know if they were photosynthetic or possibly not, date back to 1.5 to 1.8 billion years ago. Some suggest that there might be some fossils at 2.1, but in any case, something like 1 to 2 billion years after the origin of prokaryotic organisms. And there is a second type of evidence that is even stronger, suggesting or indicating that actually prokaryotes predate eukaryotes, and that's the presence of mitochondria in all eukaryotic cells, or their derivatives. So we know now that organelles such as mitochondria and the chloroplasts derive from ancient endosymbiotic bacteria. In the case of the chloroplast, it was discussed yesterday by David. These were cyanobacteria. In the case of mitochondria, we now know that they derive from a particular group of proteobacteria, the alpha proteobacteria. Right? And so we know that, and we know that all eukaryotes, the last common ancestor of eukaryotes, had mitochondria. So at the beginning, we didn't know this, and it was thought in the first molecular phylogenies of eukaryotes based in genes such as the 18S ribosomal RNA gene, dating back to the late 80s or beginning of the 90s. We had a picture like this, with the Archaea, the bacteria, and then a very long branch which contained many long branches appearing progressively, that were very long, and then an apical part that was called the eukaryotic crown, containing the plants, the animals, the fungi, and a series of other protist lineages. And many of these basal lineages were parasitic or living in, or anaerobic, corresponding to anaerobic protists that were fermenting and had kind of strange organelles like hydrogenosomes, and these lacked typical mitochondria. So the idea at that time was that these organisms that apparently lacked mitochondria had never had mitochondria, and that the mitochondrial endosymbiosis came after the divergence of this group, these three groups, that which people called at that time the Archaeozoa. However, this idea was refuted later, so we know that this is not the case, when increasing evidence started to show that there were genes of clear mitochondrial origin in the genomes of organisms of these three groups, suggesting that they had had mitochondria in the past, that they had lost them secondarily or reduced them. So this was one thing. And there was increasing evidence that, for instance, the anaerobic protists in these groups that had hydrogenosomes, in reality these hydrogenosomes were just derivatives of mitochondria. So hydrogenosomes are organelles that ferment and liberate hydrogen in some anaerobic protists. So these kind of organelles could be linked phylogenetically to mitochondria. And furthermore, in organisms that completely, apparently lacked mitochondria, people found some kind of remnants that were called mitosomes to which some proteins were of mitochondrial type were targeted back, and this could be seen by immunolabeling. Okay, so this meant that the ancestor of all known eukaryotes had mitochondria, and those lineages had lost them secondarily or reduced them or modified them secondarily. The question that remains open, and as you will see, it gives rise to different models, is whether mitochondria appear somewhere along this stem, or was really at the origin of eukaryotes at the bottom. So that's something that we will see. And another important fact that we know, this is a very important observation, is that eukaryotes, the eukaryotic genomes and eukaryotes in general, appear to have a mixed heritage with all genes and proteins involved in replication, transcription, and translation of archaeal type. This means that they have either only homologs in Archaea, or if they have also homologs in bacteria, they are much more similar to the ones in Archaea. So this is what people call informational genes, or informational genes. Whereas eukaryotes share with bacteria genes involving carbon and energy metabolism, and also, and this is very important, the nature of their membrane lipids. And we will see this later. So how to explain this kind of mixed heritage in the case of eukaryotes? At the same time, because we know that the ancestor of eukaryotes had mitochondria, we also know that symbiosis must have played a very important role in eukaryotes. And this is very important. I want to highlight the role or the capacity or the potential to explain evolutionary changes, important evolutionary changes in evolution via symbiotic interactions. Because when two organisms, let's say A and B, establish a symbiotic relationship, and this symbiotic relationship becomes obligatory, and this happens when one of the organisms exchanges or transfers genes to the other and lost some of these genes. And once one of the organisms has lost some of the genes, it depends obligatorily in the host, and the other organism in the other partner. So this implies a change of selective constraints that is applied to the consortium. And this, when you have an obligatory symbiosis, this implies that there is, among other things, gene and genome redundancy, that you have genes that encode for the same type of function in duplicates, and then many things can happen. So maybe one of the genes can be lost because you just need one to make that function, but you can also keep one gene as a backup copy that is functional and working, and the other one can be relieved of selective constraints and increase the evolutionary rate and evolve new functions. So this can lead to an increase of evolutionary rate of duplicate copies or duplicate functions, and to innovation very easily. And this could, in principle, explain an increase in complexity such as the one that is seen in eukaryotic cells. Okay? So this is something that must have happened at least in eukaryotes with the incorporation of the mitochondrial ancestor. Okay? Meaning that there were important changes leading a process that we don't know very well, leading to the last common ancestor of eukaryotes. Okay? So we don't know the details of how this process occurred, but people are trying to answer to this question, which is really the origin of eukaryotes, by looking at the patterns, among other things, looking at the patterns of gene distribution and features in general. On one hand, on extant eukaryotes, to try to define what was the last common ancestor of eukaryotes like, and to the potential, and looking to the potential distribution of these same genes and features in the mitochondria and the host ancestors. So what do we know now about this? In the case of the last common ancestor of eukaryotes, there are several labs working on this, and it is possible now to tackle this problem thanks to comparative genomics, because there are now a reasonable number of genomes at stake, and not only plant and animal genomes or parasite genomes that were the only kind of genomes that you could find a few years ago, but now people are starting to sequence genomes, eukaryotic genomes of protist groups that are free living and that could represent better the diversity of eukaryotes. So that you can compare the genes in all these genomes and make predictions about which were the genes that were common and therefore present, potentially present in the last common eukaryotic ancestor. So by doing this type of comparisons with the relatively limited data that is available, some people have tried to do that, have tried. And the image that we get is that of a very complex last common eukaryotic ancestor. So the last common ancestor of eukaryotes was something already quite complex with several thousands of genes that had a high metabolic flexibility and complexity, developed cytoskeleton, nucleus and nuclear pore system, a complex system of endomembranes with endocytic and exocytic capacity and various endomembrane-bound organelles, a eukaryotic flagellum, a proteasome, ubiquitin signaling, splicing, mitosis, meiosis, and even RNA interference. So the features that are generally widespread today in eukaryotes. And some of these cellular systems, or the evolution of such systems, can be relatively easily inferred from prokaryotic ancestors. One of the most clear is the cytoskeleton, because we know that now that both bacteria and archaea harbor homologs to tubulin, that is FTZ, and tubulin and actin, sorry, the gene, the protein MREB. There are homologous, so the primary sequence is not very well conserved, but when you look at the structure, the three-dimensional structure is really very well conserved in the two cases, and the function is also conserved. So you have polymerization of these proteins in prokaryotes, making microtubule-like structures, and the same occurs for actin. So it's reasonable to think that from this type of prokaryotic ancestral proteins, the cytoskeleton of eukaryotes evolved. It is more complex for the rest of the system. So we have seen what the last common ancestor of eukaryotes might have looked like. Then there remain questions, of course, about the mitochondrial ancestor. We know it was an alpha proteobacterium, but we don't know exactly what kind of alpha proteobacterium. Some people have linked mitochondria to the rickettsiales, which are intracellular parasites infecting many kinds of organisms, including amoeba, for instance, or to the free-living bacteria Pelagibacter, which is a cosmopolitan organism in oceans. There is free-living, but has also a reduced genome. In the two cases, actually, these two the genomes of these organisms are highly enriched in A and T, so very low GC content, and there is possibly, so the affiliation of the ancestor of mitochondria with these organisms has been questioned because there is a problem of GC bias, and this is possibly, these relationships are possibly not true. They join together just because, in phylogenetic trees, just because there is an important GC bias in mitochondria and in these two types of alpha proteobacteria. So people are working around this. And the other question that remains open in the case of in the case of mitochondria and its ancestor, was if this ancestor was a facultative anaerobe. So we know today that mitochondria perform, in most cases, oxygen respiration. So you oxidize organic molecules with oxygen, but many mitochondria that are really widespread in the eukaryotic tree can ferment, just as hydrogenosomes do. Hydrogenosomes, I recall, are organelles that are derived from mitochondria, present in many anaerobic protists, and in many mitochondria of anaerobic protists, they also can use acceptors other than oxygen, such as nitrate, fumarate. So, is this because they are widespread in the phylogenetic tree of eukaryotes? Does this mean that this capacity to use other electron acceptors was universal or was present in this ancestor? So this is an open question. But the most open question of all, and this is where most people oppose and fight even, is the nature of the host that acquired mitochondria. This is a really open question. There are many different models and an ongoing controversy. So we'll try now to summarize these different models. It's impossible to be exhaustive, but in types of models, okay? I'm focusing on those that propose more details in the process of how eukaryotes should have appeared. So the most classical model that I would call here the proto-eukaryote, or some people call the tree of life model, is as follows. So eukaryotes, from a last common ancestor, there would have been a split in two major lineages, one leading to bacteria and another leading to the ancestor of Archaea, and a third proto-eukaryotic lineage that lacked mitochondria but that developed all the features except for mitochondria that are characteristic of eukaryotes today, including the cytoskeleton, a complex endomembrane system, and so on and so forth. And at a given moment, this proto-eukaryote heterotroph incorporated the ancestor of mitochondria via endosymbiosis, and then in this type of model, the bacterial features in eukaryotic organisms would have been imported via the mitochondrial endosymbiosis secondarily. Okay? So this is the most general model. There are different variants, but this is the traditional model. There is somehow a variant of this idea is the model proposed by Tom Cavalier-Smith, the Neo-Muran hypothesis, in the sense that he proposes that eukaryotes derive from an independent proto-eukaryotic lineage that was sister to the Archaea, and that acquired mitochondria from a gram-negative bacterium by endosymbiosis quite recently. For him, it would be like 700 million years ago, so the origin of eukaryotes would be very recent. And the only difference with the previous one is that this split between Archaea, that actually this split would occur within the bacteria. So the last common ancestor was not leading to, on the one hand, bacteria and Archaea, but was already a bacterium. And in his thought, that this difference is due to the fact that explaining in bacteria you have two major types. There are the gram-negative, having two membranes, and the gram-positive, Firmicutes and actinobacteria, having just one membrane. And for him, this membrane loss must have happened only once. So for him, the last common ancestor must have had two membranes, and then there was one single loss leading to the gram-positive bacteria, the Archaea, which in general have one single membrane, but not always. There are Archaea with two membranes, so he cannot explain that. and to the eukaryotes that have one membrane. So this group would be monophyletic in his view because they contain just one membrane. Okay? His particular view. Now the idea that eukaryotes derive from within bacteria has been proposed independently based on the presence of endomembrane systems. So there is a series of models that propose that eukaryotes derive from particular types of bacteria, called, in particular the planktomycetes, because some of them, they develop endomembrane systems, some of them defining, such as Gematodes globus, defining a region that looks like a nucleus. It is not a true nucleus because there are ribosomes in the two sides of this endomembrane, but it resembles like a nucleus, and there are some, there is a single form of endocytosis. So some people have proposed that this type of bacteria would be at the origin of eukaryotes. However, some people, by making analysis of the genes in the genomes of these planktomycetes in particular, show or have shown that there is no particular gene that relates or gene content that relates the planktomycetes to eukaryotes, and that this is a case of analogy and not of homology. Okay? Actually, the formation of endomembrane systems in bacteria has been known for quite a long time. So you have here several examples of bacteria containing endomembrane systems like such as some beta proteobacteria that are oxidizing ammonium, or alpha and gamma proteobacteria that are methanotrophic, so oxidize methane. There are even the periplasmic space could be, and in most of these cases, this is related to the fact that you need to compartmentalize particular metabolisms. That's what happens in the case of cyanobacteria, which develop endomembranes, quite conspicuous endomembrane systems. And even the periplasmic space in gram-negative bacteria could be considered a metabolic compartment in this sense, surrounded by two membranes. Okay? And furthermore, the fact that some of these organisms, such as the cyanobacteria, develop internal endomembranes led to, in this case again, Cavalier-Smith, many years ago, when people still were reluctant to the idea that mitochondria were derived from symbiosis. So this capacity to form endomembrane systems led him to propose that eukaryotes derive from cyanobacteria, just because they have this type of endomembrane system. So he proposed that all the kind of membrane-bound organelles within eukaryotes were derived from thylakoids from cyanobacteria. Of course, this is not what we think today, and this is not what he thinks today, but it was proposed. So those are cases of analogy and not of homology, that is of vertical inheritance. So we have seen models of an autogenous and independent origin of eukaryotic cells. There are summarized here. So there was a common ancestor, a universal ancestor, bacteria on the one hand, and Archaea plus a proto-eukaryotic lineage on the other hand that incorporated the ancestor of mitochondria. The problem is that actually there is no evidence for the existence of a proto-eukaryotic lineage that lacked mitochondria. So we don't have evidence that this type of third lineage that had all the features of eukaryotes but mitochondria ever existed. So many people have simply think that this is not parsimonious, or that the origin of eukaryotes could be simply explained by a symbiosis directly between bacteria and Archaea, without the existence of a proto-eukaryotic lineage that predated the acquisition of endosymbionts. And here again there are different models. The first and perhaps most famous is the serial endosymbiotic theory of Lynn Margulis. So Lynn Margulis, as you may know, recovered the old ideas by Merezhkovsky, who had already proposed that the ancestor of chloroplast was a cyanobacterium, and she also proposed that mitochondria were derived from alpha proteobacteria. So this is now mainstream science. Everybody accepts this. But then she further proposed that the host that incorporated the ancestor of those organelles was itself derived from a symbiosis between a thermoplasma-like archaeon, that is a wall-less archaeon belonging to a specific particular group, the thermoplasmatales, and the spirochetes, which would have provided the motility to the assemblage and would have become flagella. Okay? This is highly debated and not many people believe in this. But then we have two other models that are actually more or less the same model, or they are based in the same principles, and the principle is the following, is that the incorporation of an alpha proteobacterium within an archaeon triggered eukaryogenesis. Okay, and this is very important. These two models are the model proposed by Denis Sersi in 1992. He proposed that there was the ancestor of mitochondria incorporated within a thermoplasma-like archaeon. So actually, Lynn Margulis picked the idea to Denis Sersi that this was a thermoplasma-like archaeon because it didn't have a cell wall, and this symbiotic interaction would be mediated by the transfer of sulfur. So this was a bacterium that was oxidizing hydrogen sulfide to sulfur, and the organism, the host, reduced the sulfur to hydrogen sulfide. So there was a sulfur cycle within the assemblage. In the case of the hydrogen hypothesis, that is very well known in general, proposed by Bill Martin and Miklos Müller in 1998, the host would be a methanogenic archaeon, and the ancestor of mitochondria, an alpha proteobacteria, and the relationship would be mediated not by sulfur transfer, but by hydrogen transfer. Okay, so another type of metabolic interaction. So these type of models were highly criticized for a long time because nobody knew prokaryotes harboring other prokaryotes as endosymbionts until we observed that in nature. So here you have an example. These are rare examples, but they exist. So this is one example of a beta proteobacterial endosymbiont within gamma proteobacterial symbiont. So it's a symbiosis within a symbiosis. And there are even cases of bacterial endosymbionts within mitochondria, like in ticks. So this occurs. The only thing that these models at the beginning didn't explain is an essential feature of eukaryotes, that is the nucleus. Okay? Only in 2006, Bill Martin and Eugene Koonin proposed a model for the origin of the nucleus, and I will come back to this later. At the same time, in 1998, and we refined this model later in 2006, we proposed an alternative model, the syntrophy hypothesis, that is based on two independent symbioses. In our case, eukaryotes would derive from the endosymbiosis of one archaeon, methanogenic archaeon, within a bacterium that would be a myxobacterium. So these are complex bacteria belonging to the delta proteobacteria. Their relationship would be based again in hydrogen transfer, so that the myxobacterium would be fermenting organics and producing hydrogen, and this hydrogen would be essential for the methanogen to make methanogenesis. And we proposed that mitochondria would derive from quite versatile alpha proteobacteria in an independent symbiosis within this consortium. Okay? We share with the hydrogen hypothesis the idea that there was a metabolic symbiosis at the origin of eukaryotes that was based on hydrogen transfer. Interspecies hydrogen transfer is a very well-known interaction in nature, especially in anaerobic and anoxic environments. And you have many examples of this, particularly between Archaea methanogens, so Archaea methanogens, and fermentative delta proteobacteria, such as so the neighbors of myxobacteria that produce hydrogen by fermentation, and these methanogenic archaea take up. And this type of interaction occurs also in the cytoplasm of some anaerobic ciliates that have hydrogenosomes instead of this type of bacteria, and that harbor also methanogens, so that they transfer hydrogen from the hydrogenosome to the methanogens. Okay, so this is just to say that this type of symbiosis is widespread in nature. Because I'm sure that you are not familiar with myxobacteria. Myxobacteria are social bacteria that have quite complex cycles that, for example, very much cycles in dictyostelids, for instance, so that can communicate, differentiate and create multicellular structures for a reproductive process, and they share a number of features that is with eukaryotes that is of interest. And some studies suggest that some phylogenomic studies suggest that at least some genes were ancestrally transferred from myxobacteria to eukaryotes, such as the fatty acid beta oxidation pathway. Okay, so just to say, I'm not saying that the syntropy hypothesis is true, but just to give you some elements. So if we sum up, because we have seen different types of models, if we sum up what we have seen that there are models that propose a proto-eukaryotic lineage, a model with two different types of symbionts, one archaeon engulfing the ancestor of mitochondria, and one type of models proposing two endosymbiosis and the endosymbiotic origin of the nucleus. Nevertheless, the most nowadays there is the most famous or the most trendy hypothesis is that proposing that the ancestor of eukaryotes was an archaeon that engulfed the ancestor of mitochondria, because it's simpler, you just have two symbionts. And there are several models, several variants of this basic type of model. Okay? But I will discuss with you that in general there are no, except for the hydrogen hypothesis that has been well described, these are variants of the model. In general, they do not propose any clear metabolic or any other driving force for the symbiosis or the endosymbiosis of the ancestor of mitochondria within the archaeon, or they are just based on some protein or gene content and phylogenetic analysis that are relatively partial, or we will see that, or are based on some aspects of cell biology, including the formation of endomembranes, for instance. And this is the case of this model that has proposed that this organism, there is a hyperthermophilic archaeon that has two membranes, so an archaeon with two membranes, and has a symbiont. This type of organism can even bud, so they can modulate and modulate the membrane in a quite wide form. They could be the ancestors of eukaryotes, just because they can modulate the membrane. So, well, this is just it's a case of analogy, as it was in the case of Plankton bisetaris, but I have to mention this as a potential alternative. Some people propose that the ancestor of the archaeon that engulfed the ancestor of mitochondria belonged to the Archaea. We heard about them the other day. So this is a new group of Archaea that was described in the last 10-20 years, widespread in oceans, mostly mesophilic, but not exclusively. And the interesting thing about this Archaea, because we have some genome sequences now, is that they harbor many genes that are present also in eukaryotes, and in particular, they harbor genes that are involved in the formation and remodelation of membrane vesicles. Okay, so some people have proposed that this type of Archaea might have been at the base, at the origin of eukaryotes, to the exclusion of other Archaea. The problem is that when you incorporate more Archaea, actually these proteins present that are typical of the eukaryotic export system are not only present in this Archaeota, but are also present in Archaeota, so the other group that they exclude, but scattered. Not all of them have these proteins. So you have them in some thermoplasmatales, in some methanol, yes, but they are not everywhere. So some people propose that the ancestor of eukaryotes, or the archaeal ancestor of eukaryotes, was a stem archaeon, and they proposed that the ancestor of Archaea was an organism already very complex that had many proteins, including the proteins involved in the escort system, in the membrane remodeling system, but many of these proteins of the escort system were lost secondarily in different lineages. Okay? So actually, by improving the taxon sampling, they see that these genes are not only present in the Archaeota here, but are also present in other Archaea, except that they have been lost in many others. So that's another proposal. So now we have more or less reviewed the different types of models. Can we test them? And the answer is that it is very difficult, because so we have, in very brief lines, three major types of models: those that propose a proto-eukaryotic lineage independent of Archaea and bacteria, engulfing the ancestor of mitochondria; those proposing directly an archaeon engulfing the ancestor of mitochondria; and those proposing a dual symbiosis with an endosymbiotic origin of the nucleus. That was an archaeon engulfed by a bacterium that also involved the ancestor of alpha proteobacteria. So in all of them, you would expect that to retrieve an alpha proteobacterial cyclone and genes of archaeal origin in all of them. Okay? Perhaps in these models, you would expect, in addition, to have genes of, for instance, myxobacterial type, or the type of bacterium that would have engulfed the future nucleus. But there are difficulties, because of course for mitochondria and chloroplast it's very easy to retrieve the ancestry, because they were recent acquisitions, especially in the case of the chloroplast, and essentially because there is a conservation of function. So mitochondria and chloroplasts behave and function today in eukaryotic cells just as their free-living relatives in nature. Okay? They make photosynthesis in the case of chloroplasts, and they respire oxygen like they do in nature. But the symbiotic ancestors of the host, this archaeon or this bacterium, is not only perhaps more ancient in some models, but especially they have experienced a drastic change of function with possibly innovation, so that the evolutionary, this implies an acceleration of evolutionary rate in the genes of the hosts, so that retrieving the ancestry, the true ancestry of the host might be much more difficult from a phylogenomic point of view. And this explains why many different studies using different archaeal and eukaryotic genomes find contradictory results. So some propose that there were 3 domains at the origins, others that there was just 2 cells involved. Some propose the eukaryotes, others the eukaryotes. So there are contradictions. So there are conflicting results. There is a problem here with all these studies. There was a limited number of genomes and also biased taxonomic sampling. So we need to improve this. But it is maybe not easy to retrieve this type of ancestry. So this is something that we need to be aware of. And finally, there are very open questions, really open questions, and I'm going to mention that. There are 2 of them. So let's say that most people favor now the hydrogen hypothesis and alike models. So models proposing an archaea that involves a bacterium. I will focus on the hydrogen hypothesis because this is the model that has been proposed with more details. Okay? So the problem is that we need really these details because the devil is in the details. You will see that now. One of the problems is the origin of the nucleus. These hypotheses, many of them don't explain the origin of the nucleus. The hydrogen hypothesis tries to do that. How is the nucleus explained? In the case of a proto-eukaryotic lineage that develops an endomembrane system in an autogenous way to develop the nucleus, there are 2 types of proposals that have been made. And I have to mention that there is some evidence from the presence of small GTPases that suggests that that there was a primary secretory role of the endomembrane system. This is work by Gaspar-Jekeli in several articles. But so the 2 types of selective forces that have been proposed for the autogenous origin of the nucleus are Tom Cavalier-Smith suggesting that the nucleus evolved to prevent chromosome sharing by cytoskeletal movements when the cytoskeleton developed. But the problem is that many organisms have open mitosis and they seem to do quite well, even if there are cytoskeletal movements. And cytoskeleton also exists in some bacteria and they seem to do quite well. Okay? And then Gaspar-Jekeli proposed that the nucleus would have emerged to prevent the formation of chimeric ribosomes in a proto-eukaryotic that didn't have a nucleus after it engulfed mitochondria. However, this is very difficult to accept because the only thing that this organism would have to do is to retain those genes in the mitochondrion. So the mitochondrion still harbors a genome and genes that need to be expressed in the mitochondrion to make the ribosomes, for instance, the ribosomal RNA, are synthesized in situ. So it would suffice to prevent the export of these genes to the nucleus. So you wouldn't have the formation of chimeric ribosomes. So this is a weak explanation. In the case of the hydrogen hypothesis, they proposed that the origin of the nucleus in 2006 evolved de novo to prevent the formation of chimeric protein synthesis after introns spread. But this is again very difficult to understand or seems like a contradiction because you cannot create a membrane after the problem has appeared. You have to have the membrane before introns appear. A single intron, so the presence of an intron would trigger the formation of aberrant proteins because in prokaryotes, and it would be the case here as well, as soon as you begin to synthesize the messenger RNA, there are proteins starting to translate. So in eukaryotes, the presence of introns, the presence of a membrane would serve for these people to uncouple transcription and translation. Transcription happening in the nucleus and translation happening in the cytoplasm. However, as soon as one intron appears, you have, if its presence is deleterious, you are dead. You cannot develop any membrane. So you have to have membranes pre-existing that. And that is exactly what the syntrophy hypothesis proposed. at the same time, or a bit before actually, that there was first a membrane with a secretory purpose that emerged, and this membrane actually would serve to transport hydrolytic enzymes synthesized by the methanogens once the genes were from the bacterium were transferred to the future nucleus, because otherwise hydrolytic enzymes would digest the cytoplasm, so you need a system to export them to the initial periplasm, and then only then, after, so the presence of this membrane in a sense favored the emergence of introns, and then once actually mitochondria really established in a stable way in the host, methanogenesis could be lost and the archaeal membrane disappeared. So this is a case, we propose that this is a case of exaptation. There was a membrane adapted from other thing, and that became the nuclear membrane, because once the membrane of the archaeum had disappeared, there was a need to uncouple transcription and translation. So this is a possible way. There may be others, but this is not a good explanation. This may be wrong, but this is not a good explanation. And then there is another problem, and this is the last problem I will mention, but it is also important and it is not taken into account, and that is a problem of the membranes. So membranes are made of phospholipids. They form layers like that, and the structure of phospholipids is universal. But as David mentioned the other day, there are two types of phospholipids in living organisms. The bacterial ones, so bacteria and eukaryotes, share the same phospholipid structures. They have glycerol-free phosphate linked by ester links to linear fatty acids on the side, but the important thing is the stereochemistry of the glycerol phosphate, whereas Archaea have the opposite stereoisomer of glycerol phosphate. So they have glycerol 1-phosphate moieties, usually bound by ether bonds and not ester, to branch isoprenoids. There are variations, but in general, this is the general structure. There is no single exception to this opposite stereochemistry of the glycerol phosphate. And this may seem trivial, but actually the enzymes involved in the synthesis of the two types of phospholipids are different. They are so different that some people propose that the ancestor, the last common ancestor of all organisms, didn't have membranes, and that membrane phospholipid synthesis was invented independently in the bacteria and in the archaea. There was a kind of two independent origin of lipids. And people like Bill Martin and Eugene Koonin proposed that the last common ancestor of bacteria, of all organisms, didn't have lipid membranes, but mineral membranes, and that, as in the case of this model, the bacterial and the archaeal lipid biosynthesis pathway was invented independently, evolved independently. Okay? So this is a very strong assumption. We think that this is not the case, and that actually the last common ancestor was able to synthesize both types of phospholipids, but the point here is just to insist on that the two types of lipids are so different, so that people like Bill Martin, who is the proponent, one of the proponents of the hydrogen hypothesis, proposed that lipids were invented independently for these two lineages. So this poses a problem now, because in the hydrogen hypothesis and all the other models that propose an archaeon as the host of the future mitochondrion, we have now an organism that has archaeal membrane, and now eukaryotes have bacterial membranes. So they need to postulate that there was a transition of membranes, we don't know why, to make from archaeal membranes bacterial-like membranes. Okay, and this remains unexplained. And by the same occasion, the membranes of the nucleus would be synthesized de novo. So in their model, there would be just vesicles that assemble spontaneously of of bacterial origin. By contrast, models, the syntropy hypothesis, but all other models that propose a bacterial ancestor and an endosymbiotic origin of the nucleus, in this case, we already have a bacterial membrane outside. So the only thing that you need to postulate is a classical endosymbiotic origin of the nuclear membrane and the loss of the membrane of the archaeal endosymbiote. So this may be wrong, but it seems from the perspective of the membrane origins seems perhaps easier than the other models. In any case, it is an open question. So in summary, models on the origin of eukaryotes. We need the details. Many people are making phylogenetic analysis. That is fine. But if you do not propose a plausible pathway for the process leading to the origin of eukaryotes, you may have problems like this one. What does explain the origin of the nucleus? What does explain the origin or the nature of the membranes in today's eukaryotes? Okay, so these are the two major questions that remain open. We need to make more phylogenomic analysis. They may not be the absolute answer, but they may help to discriminate between some of the formulated models. And I think that we need more genomes, and we need to explore nature to pick up other lineages that have not yet been sampled, both on the eukaryotic side and on the prokaryotic bacterial and archaea side. And perhaps we will never know, because this is finally a contingent event. This is something that occurred historically. It happened once, and so perhaps we will never know. But, okay, so thank you. Too tight, too theoretical, too controversial, too... Okay.
Is there, thank you for the great talk, and is there any evidence or data about phagocytosis or phagocytosis-like processes in prokaryotic organisms?
The only evidence that we don't know if it is by phagocytosis or not. The only evidence that we have is the presence of prokaryotes living within prokaryotes. This means that they enter somehow, but we don't know how it happens. We only have observed the existence of prokaryotes living within prokaryotes. So this means that if there is not phagocytosis, there is another way of entering the cell. But for instance, organisms such as this Gemmata obscuriglobus have some kind of endocytosis. So it's not perhaps as developed as in eukaryotes, but they form vesicles, small vesicles. So that exists in some prokaryotes. It is the same for that archaeon that we saw. So they bud. So there are these processes. They are not widespread, but they exist in some lineages.
Yeah, thank you. Very interesting.
You're welcome.
Any other questions?
About these symbionts of prokaryotes, are they surrounded by any kind of membrane, or by just freely laying?
I don't know if you can see that. I think that you don't see any membrane around. I'm not sure that we can see that in this. It is maybe too small, but I think that they are not surrounded by a membrane other than their own membrane. So we don't know.
You skipped them.
Yeah,
I'm afraid.
I went too far. Here. Yeah, I think you don't. Yeah, it's difficult to say. I think there is not a clear membrane around other than the own. But maybe I need to look better for later.
But no. So it means that no one observed the process of injection.
No, nobody has observed that. So these are established symbiosis that are obligatory. So they are always there. They are inherited. So they might have been a membrane in the past and it has been lost because it's something established for very long. Yes, this is not something happening all the time. So
Okay.
nobody has observed that.
No, it's clear. Thank you.
Thank you again for your lecture, and I have a question. Are there some propositions, some hypotheses about viral origin of nucleus?
Yes. I haven't mentioned that. Yes, there are some hypotheses proposing that giant viruses may have been at the origin of the eukaryotic nucleus. I call them ad hoc hypotheses because there is nothing in a viral genome that suggests, neither in the viral genome nor in the viral structure, that suggests a link, a physical link or a phylogenetic link with the origin of eukaryotes. So that has been proposed, for instance, the mimivirus again, etc. The only genes that are shared between eukaryotes and these mimiviruses, you can demonstrate that these are horizontal gene transfers from the host to the virus, and these are recent. So that is absolutely clear. You can show it by phylogenetic analysis. There are, of course, people that don't believe that, but they don't understand phylogenetic trees. That is the problem. You could, of course, propose that that has happened in the past, but we don't have any evidence for that, other than those genes. But those genes cannot be used to demonstrate that link. So I think, and the capsid has nothing to do with the membrane, so it's an ad hoc model. I think that viruses might have been very important as vehicles of horizontal gene transfers. Why? Because there might be some genes involved in, let's say, the telomerase or something, precursors, that come actually from viruses, 1, 2, etc. The mitochondrial RNA polymerase is of viral origin. So it's likely that viruses help to transfer genes from the endosymbiont to the ancestral nucleus. So there might be a lot of rearrangement. So I think that they might have contributed importantly to the making of the eukaryotic genome. But this doesn't mean that they are the origin of the nucleus. But yes, there are some models that propose that. But without a process, we need the detail of a process, because the devil is in the detail. So.
Thank you.
Looks like we ran out of questions. Thanks a lot.
So enjoy your weekend. Thank you for hosting us.
Thank you.
Чай тоже будет. Если кто-то еще хочет пару слов открытки написать, она тут лежит. We'll still have a tea in the end.